Hello, everyone, and welcome to our Headwaters Research Experience presentation night, the very first one. We are so excited to have you here today. We have about 4 presentations for you here, and I just want to say thank you so much to the Florida Department of Education. They funded 100% of this wonderful student research. I couldn't have met any of these students or helped them with their research without you, so a huge thank you goes out to the Florida Department of Education. All of these students are from Sarasota County, Florida. They've worked hard throughout the semester, and we would love for you to share any questions or comments on Facebook Live or YouTube. If you're here on the Headwaters login link, you can also ask any questions. We love to take questions from the audience. Few other announcements before we get going. If you are enjoying this and you know a student that would be really successful or really interested in this, we are currently recruiting for our winter research experience that starts January 15th. So be sure to check that out. And our very last thing I want to share with you is we have 5 first-generation low-income students from Upward Bound in Reno, Nevada that need funding to participate in our winter research experience. We are doing a donation fundraiser for them. For these 5 students, we're looking for $8,500. I am a first-generation college graduate, and an experience like this would have made a huge difference to my experience in college and beyond. So if you're feeling extra generous today, please be sure to donate to get other students into this really awesome program. I know I've had so much fun with the students this semester. So without further ado, we're going to start by introducing our first round of presenters. They had a mentor named Haley, who is awesome, and they're actually working in a group project with Annika and Kate. So I'm going to go ahead and let Haley introduce Annika and Kate, and then they'll take it from there.
Thank you, Courtney. Hello, everyone. My name is Haley, and I am a grad student at California State University in Sacramento. I'm currently pursuing a Master's of Biology concentrating in ecology, evolution, and conservation. I'm very excited and proud to introduce Annika and Kate, who are seniors at Sarasota High School. It has been an absolute pleasure mentoring them this whole semester. They are super enthusiastic and proactive, and were very able to adapt to obstacles they ran into while conducting their experiments. So I'm very excited for them to present their project to you.
Thank you. So our project is the effect of ABA exposure to Karenia brevis on breve toxin production. Just an overview of what we'll be going over today is first our introduction, which is split between red tide and phytohormones, the 2 main subjects of our project. Then we'll head into methodology, which is split in between our breve toxin extraction and our ABA extraction. And then we'll head into data figures, discussion, conclusion, acknowledgements, and any questions.
So just starting off with an introduction into red tide and harmful algae blooms. More than 7,000 pounds of dead sea life have washed up on Manatee County beaches during the 2023 red tide harmful algae bloom alone. And these events are becoming increasingly more happening increasingly, as they've started in Florida happening annually. And in Gulf states such as Texas and Louisiana, they were originally happening every 10 years. However, they've increased to happening every 3 years, killing more and more fish with every bloom.
So the majority of these harmful algae blooms and red tide blooms happening, especially here in Florida, is caused by the dinoflagellate Karenia brevis. And so what happened is that these large harmful algae blooms, also known as HABs, will be created by excess nutrients from fertilizer runoff, which is especially bad within those Gulf states as it runs off into the oceans. And so any harmful algae blooms in general are pretty bad for the environment and for those that live around them. And so for Karenia brevis, it produces the toxin breve toxin, which can cause neuroshellfish toxic poisoning, which not only kills thousands of fish each year, as Kate had mentioned, but also those who consume any seafood that contains any of this toxin can get extremely ill, hurting those that live in immediate areas, as well as harmful algae blooms, when they span across the water, the concentration of algae will stop from any light getting into the water, which can prevent any photosynthesis of plants and cause a lot of the oxygen to be depleted, causing many dead zones, hurting a lot of ecosystems.
So with these many harmful effects from red tide blooms, there's been a lot of research into what is happening and what is their cause. And so one main research article focused on if these red tide blooms may be a stress-linked response, specifically hypoosmotic stress, or a rapid decrease in salinity. And the study found that hypoosmotic stress did, in fact, increase the brevetoxin production in Crenea brevis. However, there was another study that took the same methodology and replicated the research, and they found that there was no significant correlation or link between stress and red tide blooms, meaning there really is a discrepancy, and there's still a gap on what is causing these blooms.
So something to help kind of fill that gap, or an area of research to look into, is phytohormones. And phytohormones are essentially plant hormones, or these chemical messengers that conduct a lot of different things within inside plants or even in algae. So as for phytohormones, there's several different kinds. In fact, there's dozens, as new ones are being constantly researched, which can be found in higher level plants, microalgae, and microalgae. One of those being abscisic acid, or ABA, which can regulate some level of growth within higher level plants, but also across all different types of plants and algae. It is a major stress hormone for these plants and algae.
So looking at ABA and stress specifically in higher level plants, ABA manages stress through increasing the plant's drought tolerance, or essentially increasing water retention and closing the stomata. There was a study that also looked at ABA and how it handles stress in algae, specifically a red macroalgae, and they found that it increased salt stress in a similar way through closing the stomata and increasing water retention, so the algae did not have as a significant effect from the dehydrating effect of the salt. The same study also found that ABA can affect cell dormancy.
With that being said, there's been really little research done on dinoflagellates, which are plant-like protists. So while we know that in higher level plants, there's presence of ABA, there's presence of macro and microalgae, there's a little bit of disconnect within dinoflagellates. So one study that we were able to find was detected presence of ABA in the dinoflagellate Scripsiella trochidea, in which they found that it had the genetic sequences in order to code for ABA, mainly for the role and stages of cell reproduction in the assessment stage of the growth of the bloom, which kind of opens that territory of, is ABA present in other dinoflagellates, such as red tide, and that we know that ABA is a stress hormone, so exactly what can that be linking to?
So we know that ABA is a stress hormone, and we know that brevetoxin could potentially be a stress-linked response. However, there is a gap between the connection between these two different chemicals, and if they kind of respond in accordance to one another, and one causes another. So this leads to the questions: how does varying concentrations of abscisic acid exposure to Crenea brevis algae affect brevetoxin production? As well as, since there is such limited research on ABA and dinoflagellates, does the dinoflagellate Crenea brevis contain abscisic acid? Is another one of our questions.
Next up, heading into exactly how we tested this, we started by making 3 different dilutions of our different concentrations of ABA. And so we chose 10 micromoles, 20 micromoles, and 30 micromole concentrations in order to test 3 different levels. And for each of those, they had to initially be dissolved in ethanol, but because they cannot be dissolved in water. So we had 2 different controls, and one was just our standard control of just our plain old red tide, but then the other one, we added in some ethanol just to make sure that if there was any effect on brevetoxin production in general at all, that it was because of the ethanol, like that it was not because of the ethanol, but rather because of the ABA.
Next, we took those dilutions that we made with abscisic acid, and we added them to cultures of Crenea brevis that were sampled from the lab we were working through, which was Moat Marine Laboratory. So we took those concentrations, we added them to each sample of Crenea brevis along with our ethanol and our water controls. And then finally we had one extra control of algae that was Daniela salina algae that we knew already contained ABA. So that way we could compare the ABA content in both and see if Crenea brevis has ABA. So once all of our algae were dosed, we let them sit for 48 hours to experience the effects of the treatments that we were adding to them.
Once they were done being exposed to that, we then needed to actually test for first our breve toxin. And so we needed to prepare our samples for extracting the breve toxin in order to test our samples using an ELISA kit to test for the breve toxin concentration. So we started by pipetting out each of our samples for each of our trials into centrifuge tubes and centrifuging them for 10-minute intervals 4 times at 2,500 rotations per minute. And this is in order to be able to bring everything out of the water column and collect it at the bottom. So we could just have that more purish water column at the top that we could save then for later testing using our ELISA kit. But we centrifuged at such a slow speed because we also did not want to actually break up any of the cells yet, as those were then later preserved for our actual ABA extraction procedures, in which we would break up the cells from there.
So once we had our algae samples of brevetoxin, we put them into an ELISA kit, and our specific ELISA kit had chemicals in it that would test for brevetoxin, and it would react and change a different color. And then once you pipette all of the brevetoxin samples in, you can put that ELISA kit into a microplate reader, and it will read the absorbances of light, allowing you to test the specific concentrations of brevetoxin in each algae sample.
At that point, we were ready to test for our ABA concentrations using a separate ELISA kit, so we needed to prepare our samples for that. So we started by pipetting out our little pellets of algae from our initial centrifuging of when we were originally taking the particles out of the water column for our breve toxin. And so that little bit that we preserved, we ended up freezing to help kind of starting the process of breaking up those cells, but then thawing and then using a syringe with a needle pressed against the side of the tube in order to suck up and in and out our cells to help break them up, and which we also then centrifuged them originally for 2 minutes for 13,000 rotations per minute in order to also break up those cells even further to release any ABA, if there was any ABA at all to be found, that then would be left in our supernatant that was used for testing in our ELISA kit.
So once we had a sufficient amount of abscisic acid, we followed similar ELISA kit procedures. However, this time the chemicals would test for concentration of ABA, and we followed a similar pipetting process as well as still used the same microplate reader to find the different concentrations of our samples.
So next up, we can talk about some of the results. So to get these initial data figures, we took our absorbance readings, and then using the standards, we create a standards curve in order to then convert all of our absorbances over into actual data values. So our graph to the left is on the ABA concentration in breve toxin, while the graph to the right is just showing the presence of ABA in Crenea brevis and comparing that alongside Daniela salina, our positive control algae, as well as you can see our 2 p-values, which Kate will go into soon. However, you can see in general there is just a visible decrease of breve toxin to the amount of ABA that we added.
So looking at the statistical analysis in general, we did a ANOVA test between all brevetoxin samples, and what we found was that P is less than 0.05, meaning that there was a strong decreasing trend between ABA and brevetoxin production. And so as ABA concentrations increased, the general brevetoxin production of the cell decreased, as well as Annika started to mention, there was our p-value between the positive ABA control as well as the Crenea brevis control was 0.2, And so it was less than or greater than 0.05, meaning they were statistically similar, and there was a significant amount of ABA and Corynea brevis to start. Finally, we did a t-test between the ethanol and the water controls, and this was to make sure that the ethanol had no effect on the algae's brevetoxin production, and P came back greater than 0.05, meaning that that did happen. As well as we took water quality samples of each algae sample, and what we found was that the pH stayed roughly the same. However, the dissolved oxygen percentage as well as the cell counts in each sample increased, and this means that the cultures generally became more healthy, and in a sense, the ABA de-stressed the cultures and allowed them to flourish.
And just further proving that point, looking at some visual observations here, our first trial from each of our these are images of our first trials from each of our different samples. And so looking at from starting at number 7 was our 10 micromole concentration, all the way up to our sample 13 there is our 30 micromole concentration. You can see as the water has starting to get much more clear as the health of the algae increased. So as for some conclusions that can be made, our hypothesis initially was that ABA was going to actually increase the amount of brevetoxin toxin that was released. However, that is unsupported, as it proved that there was actually less brevetoxin released. And so what this could mean is that brevetoxin is a stress-linked response, as the stress hormone actually ended up as it de-stressed the algae, it caused the algae to produce less brevetoxin, and which proves that brevetoxin must be a stress-linked response. And so as of right now, we don't know which phytohormone necessarily produces directly produces brevetoxin, but we do know that ABA is something that causes a decrease of brevetoxin production, which opens the floor for future research on which phytohormone does produce brevetoxin, if it is a phytohormone at all.
So just for some acknowledgements, we'd like to obviously thank Headwaters, our amazing mentor Haley for helping us, as well as our remote marine laboratory and ecotoxologist Jennifer Toyota, and then our teachers at school who were able to help us with our project and for allowing this to happen.
Wow, that was incredible, girls. I've loved watching your work throughout the semester, and I got a sneak peek of all these presentations last week, and I don't know how you managed to get it even better today. So congratulations, great job. I'm going to let Haley come with the first question, but I just want to remind everyone you can go ahead and put questions if you have some for our presenters on Facebook Live or on YouTube. But until then, I will let their mentor Haley start with the very first one.
Okay, well, awesome job. That was great. It was so nice to see everything put together. My question, I have a couple, but my first question is, does K. brevis naturally contain ABA? Were you able to find that out? My internet was cutting in and out, so I think I missed.
So yeah, we were able to find out that Corynea brevis does naturally contain ABA, considering that we had our control sample that was an algae that we already knew contained acetic acid, and we ran the t-test between the two, and they came back statistically similar. So we were able to actually prove that, which was cool.
Okay, and then were there any challenges that you ran into as far as in the initial stages of your experiment, and how did you combat that in case others want to replicate your project?
We definitely had many different issues at the start, mainly in the testing phase, as we initially dissolved the ABA in too much ethanol for our dilutions, which ended up killing majority of our algae early on. And so something that we kind of learned to go back and do smaller tests to make sure that we were putting not like too much ethanol in there, and decrease the amount of ethanol we used to originally dissolve the ABA to being the bare minimum that we needed for our dilutions.
Okay, awesome. Thanks.
Awesome. And I have one question for you all. And reminder, anyone in the audience, please toss a question in. These girls did really awesome research. And I think my question for you all, as I learned so much throughout the semester with you, is what do you think are some of the real world applications of your research?
Well, I think that obviously, especially in Florida, red tide is such a big issue. So I think you could really take some of this research and that focused on what is causing that to use it to find potential mitigation methods and decrease the severity of the blooms in the future.
Yeah, that was an awesome answer, Kate. Well, congratulations, Kate and Annika. You all have done such a good job this semester. You should be really proud of all the hard work you've done. So with that, I'm going to go ahead and bring them down and we'll get ready for our next presenter. But before we do, I see that we have a few people on here, so I just want to do a few plugs. Just a reminder, my name's Courtney Kudra. I'm the project manager here at Headwater Science Institute for the Research Experience. All of this wonderful research that you're seeing here is 100% funded by the Florida Department of Education. So thank you all so much for allowing me to get to know your amazing students and work with them throughout the semester. It has been so much fun. Headwaters is accepting applications for our winter research experience. So if you like what you see, you know a student that would be really successful in this, please go ahead, check that out, check out our website, and I'd love to see you in class. And then the very last thing I want to share with you in between presentations right now is we have 5 first-generation low-income students from Upward Bound that really want to participate in our winter research experience. So right now we are working to fundraise in order to get them into class so they can do awesome research like Annika and Kate's. So if you're feeling generous, please check out that tiny URL. Check out this QR code here to donate. All I want for Christmas is for these 5 students to be able to participate in class. So thank you all so much. And with that, I'm going to introduce our next presenter, which is Eden Hurley and her mentor, Jesse Scott. So Jesse's going to go ahead and introduce Eden.
Hi, my name is Jesse Scott. Please bear with me. I am a 4th-year PhD student at University of Florida. I am also a professor at St. Louis University. I teach geospatial sciences and emergency management. My professional background is in hydrology and ecology with a little bit of wolf and moose wildlife biology at Isle Royale National Park as well. I'm super proud to present Eden Hurley. She did an outstanding project on stem cells. Sorry, I've got a odd background. She did a fantastic project on stem cells, and she is a biomedical student pursuing a biomedical career in the future, and she attends Sarasota High School. So I'll let her take it from here.
Thank you. So my project is on the usage of stem cells on wounded planarian. To begin, a study in the International Journal of Surgery by Jeffrey Dobson, a researcher in a heart trauma and sepsis research laboratory, states that out of 310 million major surgeries performed each year, 1 to 4% of these patients will die from their procedure. Along with this, 15% will have serious post-operative morbidity, which are chronic issues, and 5 to 15% will be readmitted within 30 days. This accounts to an annual morbidity rate of around 8 million patients as seen on the graph. Post-operative wound infections account for the highest morbidity and mortality rates in accordance with surgeries. As seen on the Kaplan-Meier survival estimates chart, that survival probability significantly decreases once a patient obtains sepsis, which is the body's extreme response to infection. Surgical site infections create large wounds that hold high counts of bacteria. Although stem cells are cells within tissue, such as bone marrow, which are able to maintain their own populace. This allows them to reproduce into identical daughter cells, which do not need to further adapt to achieve a functional stage. Stem cells differentiate by dividing into other organisms and specializing. These cells are able to make other cells for blood, bone, muscle, and tissues. Stem cells innate immunosuppressive effects that cannot cause malignant transformations after the infusion of these cells. In addition, in a study on whether transplanted mesenchymal stem cells are effective for skin regeneration in acute cutaneous wounds of pigs, cultured bone marrow cells from the femurs of male pigs were used in testing the characteristics of growth and recovery. Incisional wounds were made in the pig's dorsal skin, then infected and after injected with mesenchymal stem cells. The wounds were analyzed and scored based upon scarring, crust, texture, orientation of collagen fibers, rate of ridge, density, and maturing. The results from this study proved that the usage of stem cells on wounds allowed them to heal quicker. In the graph shown, macroscopic scores of the wounds after 28 days of surgery, the mean score for the control group was 1.84, while the stem cell group was 3.55. The stem cell test groups had much higher scores compared to the wounds healing without any additional treatment. Furthermore, planaria is a type of flatworm that contains neoblast stem cells, which are active in muscle, serving as regenerative cell. This causes incisions in the body to not seal, but create duplicate parts instead. Neoblast stem cells are similar to embryonic stem cells because they have a large nucleus and cytoplast. All of these new somatic cells forming tissue originate from these stem cells. My gap in research is in relation to stem cells and how they can impact post-operative wounds, dealing with the way that stem cells can be used to repair damaged tissue. These cells remain in a neutral developing stage, which can specialize at any point. Stem cells have been shown to stimulate the body's healing. By studying the way that stem cells affect cuts made in planarian and their recovery time will impact the methods used to treat human patients in the real world. The use of stem cells can limit the time of healing and further lessen the rate of infectious bacteria that leads to sepsis on post-operative wounds, decreasing morbidity and mortality rates overall. My research question is, how do different concentrations of adult pluripotent stem cells impact the recovery rates in wounded planaria? I hypothesize that as the concentration of stem cells increases, then the faster the wounded planarian will grow. For example, from a study from the World Book of Surgeons, shows that the usage of platelet-rich stem cell plasma was shown to improve the rate of wound healing. This is due to the stem cells being able to evoke a healing response, giving them the ability to replace damaged cells along with cells that fight off infectious bacteria. In order to test the way that stem cells affect planarian healing, first 11 to 15 millimeter laterally cut freshwater planaria were placed in petri dishes filled with a calcium chloride solution. Then the stem cells were cultured from the planaria. In order to do this, Hohlfreder solution and trypsin solution were added to tubes and placed in distilled water that was frozen at -20 degrees Celsius with 2 filter squares on top. The planarian were diced up and added to the tubes through a pipette. This allows the cells to dissociate into single cells for incubation. The cells were then centrifuged twice and put back in the Hohlfreder solution to be filtered through again. Finally, some planaria in the calcium chloride dishes were placed in a variety of concentrations of stem cells which were added to each dish, with one control group, one half stem cell group, and one double the first amount of stem cells. The planarian were measured by length of growth each day to see the impacts of stem cells compared to the control group. In my research, I had a setback when my solutions evaporated over the weekend, killing the planaria. After this experience, I adjusted my project by only using spring water instead of calcium chloride solution. When restarting my project within the first 3 days of growth, as seen on the graph, the control group had the lowest average growth rate on the first day of 0.83 millimeters. The first day had the most significant difference, and the group with some stem cells had an average growth of 1.67 millimeters, while the stem cell group with double the amount as the original had a growth of 2.34 millimeters. There was an 87% confidence rate showing that there was some significance, but the test group was very small. As one can see, the growth rates continued to be significantly greater for the group with more stem cells. This is why the group with stem cells were able to grow back to their original length the fastest. This proves that stem cells are able to increase healing wounds in planaria. I just want to thank you, the Headwater Science Institute, and Miss Courtney for all her time that she spent with us, and my amazing mentor for all the help he gave me, and my teachers at school.
Wow, Eden, this is amazing. It was so wonderful to see your presentation again. And while I know I learned a lot, it also gives me a lot of confidence in the future of biomed, knowing that you will be a part of it. So with that, I'm going to let Jesse go ahead and start us off with any questions he may have for you. And I know I have a few as well. And anyone in the audience, please put some questions in. We'd love to hear from you.
I am extremely proud of Eden and how she managed to overcome all the challenges that were up against her. And she had to reorder equipment, and then she had to expedite resampling and learning new statistical programming software. She was extraordinary, and I know she's going to do amazing in her future. So I was very proud of her, happy to help her and be part of this journey. I have two questions. Was there a significant difference between the test group and the control group? And then I'll repeat that. Was there a significant difference between the test group and the control group? And the second question is, was the test conducted successfully to meet your research objective? So was the test conducted successfully to meet your research objective?
So there was some confidence in my test group versus control group, but the trials that we were able to do were limited to 3 days and only 3 samples per test group. So you can't necessarily have too much confidence, but when looking at the data, there is a 87% confidence rate.
Great.
Excellent.
And I'm going to put this one up now. Jesse, if you want to go ahead and repeat it.
Yes. I think the second question is, was the test conducted successfully to meet your research objective?
So my test was able to be conducted successfully the second time around, and I was able to see a difference between the stem cell group and the stem cell group that had even more than the one before compared to the control group with no added stem cells.
Awesome. I have a question for you, Eden, and I'm going to bump it up here on the banner. And it says, with unlimited time and resources, how would you enhance or change your experiment? I know that you had more time and had a few setbacks that were completely out of your control that you've explained. So I'd love to hear, if you had no limitations on time or resources, how would you change or enhance your experiment?
So if I had unlimited time, I would change the experiment by having a much larger test group and be able to know the specific amounts of stem cells that are in each one with additional technology resources, and be able to see what levels of concentration stem cells are able to increase healing rates fastest.
Wow. Well, I know we live quite a far distance apart, but I hope to someday receive care from you in the biomed field. Very confident in all the things you're going to be able to accomplish, Eden. So great, great work. I'm going to go ahead and pull you guys down off the screen. Hi, everyone, and thank you for being here. My name is Courtney Kudra. I'm the Research Experience Program Manager here at Headwaters, and you're watching the presentations from Sarasota County high school students. And I just want to thank Florida Department of Education again for 100% of the funding for this wonderful student research. I have had such an amazing time getting to know your students, learning about their research, about their passions, and just seeing how bright their futures are. And I'm going to put out another plug. If anyone is interested in supporting 5 first-generation low-income students from Upward Bound to participate in our upcoming winter research experience, please consider making a donation. I love working with the students as a first-generation student myself. I know this would have made a huge difference to me when I was in high school. So go ahead, you can scan our little QR code here. We also have the link. It's also on our webpage if you forget or want to go back later. But thank you all so much for being here today. And just a reminder, you can plug in some questions or comments into the chat as well. Without further ado, I'm going to introduce our next participant, who is Eva Poor. Eva's mentor was not able to attend today, so I'm going to be doing their introduction for them. Eva goes to Suncoast Polytechnical High School, and it's been so wonderful to get to know her throughout the semester. Not only is she an incredibly hard worker, but also really passionate about birds, which is something I happen to personally be passionate about as well. So without further ado, Eva, go ahead and take it away.
So hi, like said, my name is Eva Poor. I was mentored by Anna Richmond from the University of Nevada, Reno, and my research was on the adaptations of birds in response to phenological mismatches. So for the agenda, in the introduction, I'll explain what a phenological mismatch is and whatnot. My materials and methods, which was a meta-analysis. My results, which include all my data figures and information. Then we'll go over the discussion, acknowledgement, and questions. So what exactly is a phenological mismatch? It is when a migratory bird has the timing of its arrival based on the peak resource abundance in the area. But these timings have become unaligned as factors, often being rising temperature, cause the phrenology, or the annual timing, of peak abundance to change. This mismatch can cause major population declines for birds as they either struggle with food shortages or unable to fully care for their young, since this is especially prominent during nesting periods. This is especially problematic for long-distance migrators, as they have no cues that allow them to determine if they need to change their timing, as their primary migratory cue is photoperiod, which is the length of the day, which obviously does not change with climate. Still, there has been some adaptation to this problem, mostly attributed to phenological plasticity, that is, the individual difference in timing and the ability to change the timing without any genetic differences. Kopak and Both, two researchers very familiar with this field, identified 4 major trends of adaptation in 2002, that is, earlier spring migration, later fall departure, earlier breeding, and less migratory activity, which is, to clarify, it's either a shortened migration route or less individuals participating in migration. The question I wanted to answer was, to what extent have small, long-distance migratory birds been able to successfully adapt to changing food seasonality? I hypothesized that earlier migration would be the most effective. To answer this, I performed a meta-analysis on the 4 main adaptations, looked at their prevalence and how effective they've been in stabilizing population declines in species affected by phenological mismatches. I believe this is important, as migratory birds are an important part of multiple ecosystems, as well as serving as indicator species for the health of biodiversity as a whole. In summary, I found that earlier phenology, that is, earlier migration and earlier breeding, are most common and appear most effective, but do not fully stabilize population declines in these cases. So, to test for prevalence, I compiled a bunch of papers on proven adaptations, then combed through them using the qualifications that all included species had to be a small bird, which here was defined as a bird weighing less than 100 grams, a long-distance migrator, which is a bird that migrates more than 1,000 miles per migration, and that one of the 4 major adaptations has been statistically proven rather than assumed, as was common with earlier breeding. Once I did that, I had a sample group of 32 different species. I marked their adaptation in a table and compared it all in a bar graph. For the case studies, I chose 3 species: the black-throated blue warbler, the barn swallow, and the European pied flycatcher, mostly due for comparison's sake, due to their similar natural histories and insect-based diets, since insects are very seasonal in a lot of areas. I looked at their natural histories, their proven adaptations, and how those adaptations affected their population trends locally, and then looked at their global trends. Using that information, I graded the level of decline stabilization on a scale from 1 to 4, with 1 being no change in decline and 4 being complete stabilization. The only materials used were my computer and all the sources I got off of it, all of which were public domain. Most of them were primary source research papers, but I got some information from secondary sources like Audubon, the British Trust for Ornithology, eBird, and BirdLife International, though that was mostly natural history information and global population trends. So here you can see my graph I did for the frequency test. Earlier arrival was most common with 17 of the 32 species, then later departure with 11, and then finally earlier breeding and less migratory activity tied with 7, though it could be noted that a lot of the papers assumed earlier breeding occurred if earlier migration did, but since I couldn't include it if it was not statistically proven, the results are likely skewed to the lower side for breeding. As well, note that the numbers don't add up to 32 since some species showed multiple adaptations. For my case studies, so this is very simplified, I found blue warblers nesting in North America showed earlier migration and later departure, which helped improve their populations as they had time to double brood or re-nest if their previous nest failed, giving them a rating of 3. Barn swallows nesting in Europe showed earlier migration and shortened migration distance through latitude changes in their African wintering grounds, which didn't seem to cause very notable changes in population declines, so I gave them a rating of 2. The pied flycatchers nesting in the Netherlands showed only earlier breeding and saw a significant improvement in fledgling success as a result, giving them a 3. So it can be noted that while the warblers performed the best as they're improving both locally and globally, their populations are increasing, which is in the minority, it may be attributed in part to their more generalist diet and habitat preferences, that is, they're more varied and opportunistic. The other 2 aren't specialists by any means, but comparatively they're a bit more picky, and it's pretty well known that generalists are less affected by environmental changes since they're more willing to take advantage of other resources or areas. As well, it can also be said that there is something to be said about the dangers of certain adaptations, as in the case of the barn swallow. It was noted by researchers that if they continued to push into northern latitudes, they would approach increasingly dry desert close to the Sahara, which is suboptimal for these birds. To my knowledge, there is not any observations of this happening yet, but the negative impacts of quick adaptations could be a point of predictive research. Due to the nature of a meta-analysis, my results are obviously may be skewed by the biases of other studies or any outside factors skewing their results, as well as just the general availability of information. I also have a very small sample size due to time constraints, so it should be noted that my results are meant to be broad and will not apply to all species. I believe that my results show that while there is notable improvement in population declines following adaptive behavior, none of these studied species actually fully recovered, so in an increasingly warm planet, conservation efforts may be necessary to preserve long-distance migrators, especially species that struggle to quickly adapt or show ineffective adaptations, whether the conservation efforts be species-specific programs or general protections on ecosystems. There's still a lot to be looked at. There's much we can learn from looking more intensely at specific adaptations in certain species or unusual adaptations. And further, my results and the worrying global population trend show there's a great need for effective conservation programs and all the research that comes with that in order to safeguard our birds and biodiversity. For my acknowledgements, I'd just like to obviously thank my mentor, Ayanna Richmond, for helping me with this project and talking about birds with me. And I'd also like to thank my AP Environmental Science teacher, Ms. Henderson, for both telling me about this program and just helping me go through ideas of what question I wanted to do. So, any questions?
Awesome job, Eva. That was amazing. As a fellow bird nerd, I think that's really wonderful. I really enjoyed seeing your project throughout the semester. So my first question for you is, how do you think platforms encouraging community or citizen science could impact further research similar to yours? And if you don't quite understand some of that wording, go ahead and ask for it.
I understand that it's
Great.
like eBird and whatnot, where they have citizens take pictures and report like locations. Yes, I think it could open a lot more opportunity for research, since you don't need to hire big teams of researchers to go out and collect all this data when you can get it from citizen science. Obviously, it will be a little less reliable compared to like professionals, but there's still a lot you could do with it, and it opens a lot of opportunities, I think.
Yeah, that is a wonderful answer to your question. And my next question is, what has inspired you to pursue this specific topic? Like, what really got you into wanting to study birds?
Well, initially, I got into wildlife in general. Again, and thanks to my environmental teacher, Miss Henderson, because I did Envirothon, which is just like a natural science based competition, and there's a bunch of different sections that you get to do, and my assignment in my team was the wildlife section. So that got me initially into like the science of animals and whatnot, and then I just focused specifically on birds just because I always had a softer spot for them. It started just because I really liked ravens, corvids in general. Everyone loves corvids. They're smart.
Right.
uh and also just because I like animals that I can frequently interact with. Like if you really like bears, that's really nice, but you don't see bears that often. But I can go outside and just find a bird right now.
No, you're totally right. That is totally true. Anyone can watch birds from anywhere in the world. So my last question for you today is, how do you think changing bird migration impacts other species or ecological niches?
Well, obviously these birds play roles just like any other animal in these ecological niches. So if they change their migration route, then there will be a lack of predator for insects or whatever they eat in there, and there will also be lack of prey for whatever goes after the birds and the eggs, and then also just them spreading seeds and all that. So basically everything that birds do, everything that all animals do, affect other animals. So if they're not there, whether because their populations have declined or because they've had to change their routes due to climate or whatnot, it'll leave a mark no matter how big the animal is.
You're exactly right, Eva. Well, great job. That was amazing. It was so much fun to hear about your bird research. I'm going to go ahead and take you down and just do one last, another plug for All That I Want for Christmas is to have these 5 low-income students, first-generation low-income students participate in our winter research experience. We're fundraising $8,500 for these students to be able to participate, just like these students here were able to participate. So if you're willing, please make a donation to help sponsor these students to participate in our upcoming winter research experience. Up next, we have Jackie Williams, and Jackie will be introduced by her mentor, Jonah. And I'm going to go ahead and let him do her introduction. So go ahead and take it away, Jonah.
All right. Thank you very much. I'm Jonah. I am a PhD student at Stony Brook University in New York. I study marine ecology. And this semester, it's been my pleasure to be working with Jackie Williams on her coral growth project. It's been a pleasure for a lot of reasons, but mostly for her self-motivation and enthusiasm about this topic. She reached out and got herself a tour and interview with the staff at Moat Marine Lab in her area. For those of you who don't know, it's one of the top marine labs in the country. So she has just been fantastic all around. And yeah, take it away, Jackie.
Thank you. Yeah, so essentially my project was on optimizing coral growth and evaluating nutrient enrichment impacts. And all the photos that I've used in this presentation are from my initial interview with Moat Aquarium, and because they allowed me to use these photos for my presentation, so I would like to thank them for that. So overall, coral reefs host a rich, diverse community, fostering numerous symbiotic relationships and support humans through fisheries, food supplies, tourism, and etc. And they act as a vital ecosystem for a variety of aquatic animals, and these vital ecosystems face ongoing challenges due to continuous development and persistent pressures of climate change. Anthropogenic activity, along with other factors, are the primary cause of ocean acidification, thermal increase and like runoff in greenhouse gases. So what I was essentially studying was to what extent does the type of nutrient enrichment increase the positive yield for corals. And initially my hypothesis was nitrate will be the most beneficial in giving them the best growth. So it is important to know that corals are an indicator species as well as serve for other purposes like previously stated. They help to see how the environment is around them, and they are very important to our world. So my experimental method was essentially I was done through a meta-analysis, and I collected a series of documents, I logged them, and I researched and looked further into them. I applied them, and that's how I got my results. So within physical restoration efforts, nutrient enrichment emerges as a supportive approach, enhancing coral resilience by strengthening their adaptability. And this study performed was using past research to determine the most effective environmental addition for achieving the highest growth yields. And a gathering set of randomly selected enrichment documents examined the graded present system. The results of the study will be presented now. So the results of the study was ammonium enrichment being the most beneficial for coral growth and health, and showcasing the average of approximately 2.44% of all of what I chose to collect, which was a series of ammonium, nitrate, phosphorus, peptone, and like other fertilizers. And this really was, there's a very limited number of what I could find from my documents. So it's not exactly the most applicable, but from what I found that ammonium addition did increase the growth yield the best, even though it is a limited addition. So within my data figures, you can kind of see I had a box and whiskers plot, and right up here is where ammonium lies. And as you can see, it is benefiting the highest. And just to kind of show how their results can vary, I have marked on this map that I hand drawn all the locations from all the articles that I had researched, where they had gotten the corals and where they had their like research facilities, like roughly. So it's coming from a variety around the world. So different parts of the world may yield a better result to a different enrichment is what I found. So here's some more images from when I went to the Moat Aquarium. I found this very nice. It was like this observation tank, and unfortunately I was unable to go into the lab. However, I was able to see some really cool technologies and get some cool interviews. And addressing my hypothesis, nitrate actually proved to be the worst addition. Actually, going back, it yielded quite the opposite, and it was yielding negative results. It was killing the coral rather than helping it grow. So some other factors that could influence this growth yield could be temperature, area, and the time. So if it's in the time of the year, if it's cooler around there, which could affect the temperature. And then once again said the area. And the significance of this study was more for trying to help improve the relationship to coral and see if they can find stuff for improving their availability in the future. And through nutrient enrichment, we can help to focus our studies to help explore other ways to help them. And some remaining questions would be really, if I were to actually be able to produce this experiment myself, how applicable would this be in a real world situation if this could be taken and applied, and see if this would help in a natural environment. So that's what I would personally love to do. So essentially I want to go back over my procedure. All I did was collect a series of documents, which is I which is over probably over 40 documents, and then I determined whether or not they could work for my study, because it was very difficult to find previous documents that were working towards my question. And I logged the studies into a table, and I found 10 applicable studies that worked, and then I used those overall in comparison to everything else. And overall, I would like to thank Headwaters, as well as my mentor, my parents, and then once again, the AP environmental teacher at my school who introduced me to this program.
Awesome. Great job, Jackie. It was wonderful to hear about all your important research into corals. I know I've learned a lot and have been really inspired by your research throughout the semester. I'll let Jonah go ahead and kick us off with the first question.
Yeah, awesome, Jackie. Thank you very much. I guess my question for you, my biggest question is, now that you've done all this research, done all this reading, how do you feel about the state of coral in the world? Are you optimistic? Are you concerned? Yeah, what do you think?
Well, I am optimistic, especially since we are finding new ways to help with them, with their growth and such. So as long as we keep studying, I think we'll be in a way for our future.
Great. Jonah, do you have another question for Jackie?
No, great job.
Great job, Jackie. We actually have a question from the audience from Jack. He says, did you get a feeling for the supportive versus detrimental effects of ammonium nitrate on corals?
Ammonium nitrate. So essentially, I know ammonium had the best effect. Nitrate was not the best for corals, and it was more supportive, because from what I understand is that ammonium is actually essential for the growth of coral, but nitrate, you can't have an overabundance of nitrate, because it will cause, nitrate is also very important for corals growth, but if an overabundance of it will kill them off.
Yeah, no, that's a great answer, Jackie. I think it's been really interesting to hear about your research. And I think my last question for you is there anything through your research, what you've learned, is there anything happening on land that you think is significantly impacting corals, or like one specific thing you can point to?
Yeah, probably the extraction of like fossil fuels and such like that, because it's through the different types of ways, because where there's like fracking and all this type of runoff and it's greenhouse gases, it seems to be a significant contributor to what's happening with corals.
Awesome. Well, great work, Jackie. Thank you for your mentorship, Jonah. It was so fascinating to learn about corals. I know I've been inspired from a few recent popular documentaries, and I can't wait to see what you continue into the future, Jackie. So please work to save our corals, because I know that's work I would love to do and love to see you pursuing. So we just have one final presenter for us this evening. Thank you all for joining us here. I'm just going to do a few plugs real quick. A big thank you for the Florida Department of Education for funding 100% of this wonderful student research. It's been wonderful to have your students in class to learn so much about all these different important subjects that they have. If you like what you see and you know a student who'd be interested in participating, we are accepting applicants for the winter research experience. You can check out that tiny URL. It's also on our website, so if you or someone you know would be really interested in the research experience, please encourage them to go ahead and check it out. And the last thing that I have for us is we have 5 first-generation low-income college-bound students from Upward Bound that need funding to participate in our winter research experience. All I want for Christmas is that these 5 students can be in my class. So please consider making a donation to sponsor these students at this tiny URL, or check out our website for more donations. We also have our little scan code here. So please consider donating in order to benefit these 5 first-generation students. I myself am a first-generation college student, and a program like this would have made all the world of difference. So please consider donating. And we have our very last presenter here that I'm going to bring up, Sarah Gutierrez and her mentor, Jen Hepner. Jen is a wonderful mentor for us and has been mentoring for us for quite some time, is super knowledgeable. And I just want to say a huge thank you to her and all of her important work. So Jen, if you want to go ahead and introduce Sarah, I'll let you two take it away.
Hi, everyone. Thank you all so much for coming, by the way. My name is Jennifer Hepner. I am a PhD student, a PhD candidate at the University of Nevada, Reno, and I study the anthropogenic effects on urban wildlife, or specifically birds, in Reno, Nevada. I've had the great pleasure in mentoring Sarah Gutierrez, who is a sophomore at Sarasota High School. She came into this experience with not only an extremely interesting medical-based question, but also an incredible determination and motivation to answer this really difficult question. Even through all these research setbacks with lab space and materials, she has stuck through it all. So I'm really excited for you all to learn from her vast knowledge on this subject. So please enjoy Sarah's presentation, everyone.
Thank you. So my project is essentially on the comparative effect of differing minerals, more specifically pyrite, smectite, copper, and silver, on gram-positive Staphylococcus aureus and gram-negative Escherichia coli. So for my agenda, today I'm going to be going over some introduction, background, methods, my predicted results and data figures, discussion, acknowledgements, and then any questions. So bacterial infections are incredibly dangerous, as demonstrated by the fact that they are actually the second leading cause of death worldwide as of 2019. And silver's bactericidal properties are often used as a treatment against bacterial infections. Pyrite, smectite, and copper are all other minerals besides silver known to possess antimicrobial properties. And my experiment aims to fill the gap in research in that pyrite, smectite, and copper's potential applications in wound infection treatment are currently unknown. So any ailment resulting from bacterial growth is a bacterial infection, and these infections are incredibly prevalent in post-operative wounds. In this area, they have incredibly high morbidity and mortality rates and are therefore even more dangerous. Staphylococcus aureus is a bacterium often found within the nose, and Escherichia coli is a bacterium often found within the bowels. However, if these bacteria enter the body through unprotected bodily passages or grow in other locations, they have the potential to cause deadly infections. These are the 4 minerals I will be using in my project, so they all have some sort of antimicrobial reason behind me using them. Silver dressings have been used since the early 1970s to both prevent and treat bacterial infections, and more recently, silver nanoparticles have been tested in a wide range of studies as they have numerous mechanisms of action. Copper was registered as an antimicrobial touch surface all the way back in 2008 and is known to have biocidal properties effective against a wide range of microbes. In one proof of concept experiment for use in water treatment, pyrite killed 99.9% of waterborne Escherichia coli bacteria in less than 4 hours and is known to provide clay with its antibacterial properties along with smectite. So the question I aim to answer is there a significant variation in the degree of antimicrobial properties exhibited by pyrite, smectite, copper, and silver when used to treat Staphylococcus aureus and Escherichia coli? So my hypothesis was based on a few main facts, the first of which is that silver is a transition metal, and transition metals are all known to have similar properties. So silver also is known to possess antimicrobial properties and is effective against both Escherichia coli and Staphylococcus aureus growth. Pyrite and copper also are or contain transition metals, while smectite does not. Because of this, I hypothesized that pyrite and copper will maintain similar antimicrobial efficacy against the bacteria in my study, and smectite will differ in efficacy against bacteria. So as for my methods, when organizing my methods, you'll probably notice that I organize them rather broadly, so some steps will likely take place over a few days. So I'll start by liquid culturing my bacteria, and to do so, I will be transferring tryptic soy broth into each of my test tubes along with its respective bacteria to reach a 0.5 McFarland standard, and then I'll allow those to culture overnight. Next, on the same day, I will begin testing for mineral contamination. by transferring tea soy broth into each of 20 test tubes and adding each test tube's respective mineral powder into it. Then I'll incubate those overnight before streaking the contents of each onto their respective blood agar plate, which will be incubated once again before I will count the number of CFUs or colony forming units, if any, to record in my data table. And next, could you go back, please? Next, I will be collecting data on bacterium prior to treatment. And to do so, I'll streak the bacteria from each culture onto its respective plate and incubate the culture overnight before counting and recording the number of colony forming units. After this, I will be treating my liquid cultures, and in order to do so, I will simply transfer each culture's designated mineral powder into the test tubes, and the amount transferred will vary depending on the type of bacteria. It would be 850 micrograms for Escherichia coli and 6,250 micrograms for Staphylococcus aureus. Then I will incubate those cultures overnight, starting data collection on bacterium post-treatment. For this, I will streak the bacteria onto their respective plates to be incubated overnight before I will count and record the number of colony-forming units present on each plate. And for data analysis, I'm first going to subtract these values. So I'll subtract the value for the number of CFUs post-treatment from the ones prior to treatment in order to find the difference in number of CFUs. And then I will be using an ANOVA test to compare the two. So these are my predicted results. And a few issues occurred in preparation for my experiment that prevented me from having collected all of my data at this point. So I used information from my background research in order to generate the predicted results displayed here. And this graph is essentially showing the difference in number of colony forming units of each bacteria after treatment by each mineral. This illustrates the amount of bacteria that would be killed by each mineral in a numerical way, and it's based on prediction, of course. But as for what each value means, each color represents a different mineral, and the larger the bar, it translates to the more effective the mineral is as an antimicrobial. So just a few examples of how I got these values is I used a combination of percentages and bacterial targets, so whether they were more effective against gram-positive or negative bacteria, or if I could find information on the specific bacteria, I would use that. So for a start, silver, which is in blue, this was the most effective, known to be the most effective mineral. It's already in use for a reason. So I started with that, and I used percentages I found online and knowledge that it worked slightly better against E. coli than staph in order to create a numerical value and record it in the graph here. Then I had a pyrite, and there wasn't very much information on pyrite's relationship with staph, so I acted based on the assumption that it worked more effectively against against E. coli, as it worked incredibly well against E. coli in the proof of concept. Copper, there was minimal information, but still enough that I was able to make the assumption displayed. And smectite, there was very little information and almost none, so I was unable to make a very knowledgeable assumption and kind of had to take a guess on that. So I put it about out of the same for both bacterium because of that. And then the control, you'll notice there isn't a control bar. That isn't because there isn't a control. It's just because if you were to treat something with nothing, it's only logical that it would have no difference in number of colony forming units post-treatment. So as for my discussion, the reason I was unable to complete my results mainly consisted of timing issues in finding lab space. This took a long period of time and also required me to change my procedures significantly, which ultimately did improve my experiment, but did take enough time that I was unable to finish, along with submitting paperwork, which took a long time to be processed and approved. However, the good news is that I am starting this experiment tomorrow, so it will be finished by the end of the week. What I would change if I did this experiment again is I would work on my paperwork earlier and I would order materials sooner just to prevent any delay there. And other questions. So if my experiment does prove that some minerals are similar in efficacy to silver, I would ask the questions, Does mineral toxicity render the effective mineral unsafe for use? And if not, what quantity of the mineral can be used without endangering the patient? As for my acknowledgements, I would like to thank my mentor, Jennifer Hepner, and my teachers and parents. And this is just my works cited.
Awesome. Well, great job, Sarah. This was so wonderful to learn about. I've loved hearing your research throughout the semester, and I'll let Jen start us off with her first questions for you.
Yeah, great job, Sarah. So congrats on being able to start your experiment tomorrow. So if you are able to find antimicrobial properties in these minerals that have never been tested before, how do you think we can see them used in medical practices in the future?
So in the future, if we did find that these possess antimicrobial properties, of course, there would be a lot of other testing to go into it to assure the minerals were even safe for human contact, which is why I mentioned the toxicity studies. But you could see it used in post-operative wound infection treatment. Currently, that's the area where silver is most used. And if we were to do so, it would provide an alternative, which would allow not only to decide based on the amount available for use, but also it would allow you to choose the best for each situation. So if someone's allergic to something, or if it's one mineral is ineffective, you can try multiple others.
Awesome. And how did you become interested in this topic in the first place?
I read an article about cross-contamination within natural elements and how it had led to antimicrobial resistance, and I was curious about how other natural elements would be useful in wound infection treatment as antimicrobials.
Awesome.
Wow. Those are some awesome answers to some tough questions, Sarah. Jen, thanks for asking some questions. If any of our audience members have any questions for Sarah, please go ahead and toss those in the chat. But in the meantime, I have a question for Sarah. Actually, I'm going to let one of our audience members ask their question. So this is from Jason, and he says, how could you define what a single CFU was?
So a colony forming unit, I said count a lot. Essentially what you do is when you're looking at the Petri dish, you'll see the number of colonies, and a single colony represents 100 colony forming units per milliliter of liquid. So I multiply every value by 100 in order to find the number of colony forming units.
Wow. That's awesome. Well, great answer, Sarah. Thank you for your wonderful presentation. Thank you, Jen, for your wonderful mentorship. And we're just going to wrap up real quick. So with a few last thank yous to the Florida Department of Education for funding this program. It's been amazing to get to know your students here in Sarasota County. I know I've had a wonderful time. They're so smart, driven, passionate individuals, and I'm so thankful to have been a part of their research experience. Another few plugs is if you like what you see and you are a student would like to do something like this, research like Sarah, Jackie, Eva, Eden's, Annika, or Kate's, please go ahead and check out our winter research experience. We do have some space left, but they do fill up quick, so be sure to check that out. And lastly, I'll just say my Christmas wish list one more time. If I could get one thing for Christmas this year, it would be to fund these 5 first-generation college-bound students in our winter research experience. I myself am a first-generation college student, and I think an experience like this would have made a huge impact to my high school and college experience. So please, if you're feeling generous, go ahead and scan this QR code or check out the link below to donate. It would be so wonderful. We're looking for $8,500 to sponsor these 5 kids from within Reno. And with that, I just want to thank everyone for participating, for asking questions, for attending today. Without any of you, my job would not be nearly as fulfilling. So thank you all so much, and we look forward to seeing you at tomorrow and Wednesday's presentations. Thanks, everyone.